Assalamu alaikum and good everyone. Good evening everyone. This is me Abdul Moazim, a biotechnology student from the Quaid-i-Azam University Islamabad. And today we are going to discuss about the most devastating and most renowned topic, and that is CRISPR-Cas9 and beyond its applications. Imagine if your DNA was a giant Microsoft Word document filled with typos that causes genetic diseases. Now, what if I told you that we finally have the ultimate find and replace tool to fix those typos permanently? Isn't it seems to be excited? Let's begin. In a document, if we suspect we have misspelled a word, what we do is directly goes to the search and replace bar and correct that word, right? And the same mechanism works in our human body with a technique called CRISPR-Cas9. What is CRISPR? CRISPR stands for clustered regularly interspaced short palindromic repeats. It contains two main components, that is Cas9, which cuts the DNA, and the guide RNA, which recognizes the particular sequence of DNA. Correspondingly, before it begins, what exactly scientists do? Scientists first recognize the particular sequence of DNA that causes the genetic diseases, and then they introduce the guide RNA, which recognizes the A's, G's, T's, C's of DNA in the human body. And then this guided RNA is introduced into the target cells for the further modification. Like they locate the particular sequences of the DNA and they cut the DNA and the remaining sequence of gene, the particular short form of gene that causes the genetic diseases, and the scientists can edit the gene via either insertion, deletion, and the final product is ready. So, in somewhere down the line on the future applications, this CRISPR-Cas9 is going to cure the inherited diseases for a lifetime that couldn't even done in the our rest of laurels. The next is I've told you already about that. The CRISPR is a revolutionary gene editing technology that works like a molecular scissors. I hope so. From the previous video, you have understood what exactly the CRISPR is. Now we are gonna move towards the practical applications or the medical application, or we'll talk about agriculture phenomena and many other products that actually uses this technique. The first one is medical applications. For decades, treating a genetic disease was like constantly mopping up the floor while the faucet was still running. CRISPR lets us finally turn off the faucet. In medicine, this is not science fiction anymore, it's a reality. Take sickle cell anemia or cystic fibrosis. Instead of managing symptoms for a lifetime with endless medication, CRISPR targets the root genetic mutation to offer potential one time cures. Furthermore, in cancer therapy, we are using gene editing to supercharge our own immune cells, like uh, upgrading an ordinary police force into an elite tumor targeting squad via enhanced CAR-T cell therapies. The next one is agriculture revolution. In a world facing rapid climate change, what if crops did not just survive droughts but actually learn to thrive in them? It is really gonna be excited that how it's actually gonna be works. The impact of CRISPR on agriculture is nothing short of revolutionary. Traditional breeding that takes decades of trial and error, like shuffling a whole deck of cards just to like find a single S, right? So CRISPR lets us pull the S right out of the deck. Scientists have successfully engineered drought resistant crops that thrive with 20% less water, develop plants naturally resistant to devastating pests to slash chemical pesticide used, and created nutritionally enhanced crops. This is how we build a sustainable food supply for a growing global population. The next one is research distribution. If you look at this pie chart, It looks like a clear picture of where our scientific energy is going. But wait, there is a massive critical mistake hidden right in plain sight on this slide. Can anyone spot it? If anyone of you, then kindly comment on the below session. So I will let this exactly know. Let's look closely at the data. The text correctly states that medical research leads at 45%, focusing on the therapeutic applications like treating genetic disorders. Agriculture applications take up 35%, and basic science comprises 20%. But if you look at the Canva pie chart graphic on the right, it claims item 1 is 62.5%. This is a classic visual mismatch. If we look past the graphic glitch to the actual numbers, it shows that while saving human lives through medicine takes the biggest piece of the pie, we are investing heavily across the board. We are not just focusing on cures, we are building the fundamental biological framework for a healthier planet. Beyond CRISPR. As amazing as CRISPR is, it cuts the DNA double helix completely, which can sometimes be like using a chainsaw for a delicate carpentry job. What if we could upgrade to a laser scalpel? The laser scalpel has a ride and it takes us beyond CRISPR. Beyond CRISPR, like next generation tools like base editing allows us to cleanly change a single letter typo in DNA without even cutting the double helix. Prime editing acts a true search and replace word processor for genetic sequences. And we also have some epigenetic modification, which lets us turn a gene's volume knob up or down without changing the underlying DNA code at all. So these advances represent about 20% of current research focus, paving the way for incredibly precise treatments. The next is the key challenges. With great power comes greater responsibility. Just because we can rewrite the code of life, does it mean we always could or should? We cannot talk about the future without addressing the roadblocks. Editing human germline cells means changes are passed down to future generations, raising profound ethical questions, which is why only 20% of surveyed scientists support unrestricted modifications. On top of that, global regulatory framework are fragmented, and only about 20% of consumers currently feel completely comfortable with gene additive products. To move forward, we do not need just brilliant scientists in the lab. We need transparent dialogue, education, and bulletproof ethical boundaries to earn public trust. Thank you so much. If you have any questions or any query, just drop the comment below, and I will definitely gonna reply you whenever I got some free slot. And I would be happy if you guys are particularly or actively engaged in this session. Thank you so much once again. Have a nice day.